Okay, Cristina, so if you want, we can make a start now and people
Sure.
will join as they connect. So good morning, everyone. Well, good afternoon already. It's a pleasure to have you in our webinar from our Environmental Technology and Bioelectrochemistry Working Group. Today we have Dr. Cristina Cordas, who has accepted our invitation. She is from the Laboratorio Asociado para Química Verde, which is it's part of the network of chemistry and technology in Portugal. So they have a research center when they work in sustainable chemistry as a key component of our sustainable development. So Cristina had extensive experience in the research of bioelectrochemical systems as well as biotechnology application and fundamental chemistry. It is an honor for us to welcome you, Cristina. Thank you for accepting the presentation. And without anything further to do, please feel free to, if you have any questions, just write it in the chat, or we will have the opportunity at the end of the webinar. Thank you.
Thank you very much, Sharon, for the invitation and to all the network, of course. So I'm going to share my, okay. Okay, so can you see it okay? So I'm going to talk a little bit on the interfacial bioelectrochemistry, in particular I'm going to talk about the enzymatic systems involving oxide nitride and CO2 reduction and also hydrogen evolution. So these are some of the enzymes with which we work with. We have many more, but I think the enzymes that catalyze these reactions are particularly interesting for applications. So that's what I'm going to talk a little bit today. So my presentation is going to be divided by a small introduction, some information about the metalloenzymes, metallurgy, some key results, some possible applications. Some of them we are already engaged to, others are only things that we think can be useful, some publications on the team and future directions, but it won't take long, so don't worry. So starting with the introduction. So why bioelectrochemistry? Okay, I think everybody is a little bit engaged in electrochemistry and bioelectrochemistry, but bioelectrochemistry is really a very powerful tool because of course we with electrochemistry we can exploit this type of biological systems with entire bacteria or with enzymes, purified enzymes, and the direct coupling of these biological species to electrochemical systems are really a high advantage for us because the enzymes resulting from the evolution of millions of years, they really use very mild conditions, and without them, of course, life on Earth wouldn't be possible. And we can take advantage of that for our for implementation of new systems, of new solutions, and the mitigation of a lot of problems that our industrial activity actually has reached. So I'm going to talk a little bit on the NO reduction, as I said, CO2 reduction, and H2 evolution. This last one quite search at the moment for the clean transition of energy. So the enzymes that I chose for this webinar today is the nitric oxide reductase, the formate dehydrogenase, and the hydrogenase, nickel iron hydrogenase. So the nitric oxide reductase, or NOR, NOR, the enzyme catalyzes the selective reduction of NO to N2O. And it's quite interesting because this is an enzyme that not only does the reduction, but this reduction implies the production of an N double bond, which is quite interesting and not very often seen in nature. It has a binuclear iron active site that I'm going to show next. Is highly specific, and although this is an enzyme that is related with the nitrification pathway, it operates in a macroaerobic environment, which is really interesting for applications. The formate dehydrogenase reduces CO2 to formate, and it's quite searched nowadays for forming this add value product, which is the formate, and of course as a way of mitigation of CO2. And then we have hydrogenase that has high turnover frequency, so it's a very, very efficient enzyme, and that produces hydrogen, and it's present basically in every bacteria, prokaryote, and some eukaryotes too. And the mastering the hydrogenase is like a very interesting route. Very interesting route as an alternative route for the green hydrogen society, or the hydrogen society that some aim to be the future for the energy transition. So starting with the NOR enzyme, this is a membrane-bound enzyme, and actually, this is an anaerobic, as I said, enzyme involved in the nitrification pathway. So we have the nitrite reductase that reduces nitrate to nitrite, and then we have nitrite reductase that reduces to NO, and then we have here the nitric oxide reductase that transforms into NO into N2O, and then nitrous oxide reductase to N2. These are especially important, of course, for agriculture and soils, but it can have other uses. For instance, NO, as you may know, is also a signaling molecule in our bodies. So NO and N2O are quite important. Also, NO and H2O are greenhouse effect gases. So all these mechanism is important in general, not only for agriculture, but also to medicine and so on. If you see this scheme here on the bottom, you see that NOR has two subunits. One is in the membrane, in the internal membrane, and the other is in the periplasm. So it's here where electrons enter the enzyme. So the chemical reaction is this one, and you see the potential that in nature this occurs. It's a huge potential. But within our bioelectrochemical systems, this is reduced to basically some some 100 or 200 overpotential. So it's very, very easy to occur. So we are going to talk about, there are three classes, but we're going to talk about the C-nor from Marinobacter nauticus. So the nor has, as I told you, there are three types. This the C-nor, that is the one that we work with, and is characterized by these two subunits. This one, that is nor C, and this is nor B subunit. So here, the nor C has a cytochrome C, and it receives electrons from small molecules like cytochrome C from other species, copredoxin, and also inorganic molecules, or an electrode, of course. And then it is transferred to this B. And in this B, we have cytochrome B, IMB, IMB is not cytochrome, IMB, IMB3, and a very unusual active center, which is composed by the IMB3 and a non IM iron, which is called IMP. So this is what we are going to explore. The others are more or less the same, but for instance, in the quinol, the electrons are given by quinols, that in the quinol pool, and it's a unique unit, say it's unique protein or no subunits. And the key copper nor is equal to units, but this one, instead of IM, it has a copper A center. These are all very alike the cytochrome C oxidase, and they are actually from the same family. With the differences, of course, on here the copper and here the iron B. So in this case, two NO molecules will bind at the active site, which is this one, is the B3 and the iron B. And the way as it is going to be done, it has been a motive of discussion for a long time. So at 2006, we have started to this research on bioelectrochemistry of the C-nor. It was already studied, but only by potentiometric methods. And the potentiometric methods could not really show all the electron transfer processes that this protein contains. First, because the processes from the IMB3 and iron B can be observed individually or when they are linked together. And in potentiometry, many times we could not reach the reduction force that is necessary to see that RNB, and as so all the processes were basically merged in one value, which was around 60 millivolts. And then by electrochemistry, we could deconvolute all these redox processes, and we could see that iron B is actually minus 369 millivolts, in B3 minus 162, and the others. So this in C, which is in the subunit Nor C, the one that is in the periplasm, is the entrance of the electrons in the enzyme, and that's why it has this high value. So because, as you know, the higher the reduction potential, the higher the possibility to be reduced. So it makes a pathway in which the electron enters the EMC, and then it's going to be subsequently transferred to all the other centers. So this has a high electron transfer rate. For instance, these results that was obtained in 2006 was only possible to see at higher scan rates with cyclic voltammetry. In this case, in this figure, it was 5 volts per second. And after the determination of all the constants, the electron transfer constant rates, we actually see that these other processes that are involved in the pathway of the electron pathway that belongs to these different centers, this could only be seen at very small and low scan rates, meaning that the protein has a very high electron transfer concentrate in the active center, which is the coupling of the B3 heme and the iron B, inorganic iron B. And the other centers have a very much lower constant rate, which are exactly needed for the transfer from the electrons from the physiological partner. And so it was possible for us not only to observe all the transfer rates, but also to see, I'm not showing here, the peroxynitrite formation in the presence of O2, which is very fast reaction than the peroxynitrite disappears. So we also were able to cut the enzyme in two. So as you may remember, we have these two, the NOR-C and NOR-B subunits, and we cut it in two and purified them alone. And what we were able to see is that the center itself, meaning the NOR-B, has a different potential with when NOR-C is present or not. And that means that this is a synergetic effect of both subunits, and meaning that actually it's not really possible to have only the center, the EMB3 and the iron B together and do the same enzymatic and have the same enzymatic activity. So in this case, this is attained with direct electron transfer, meaning that the only thing that we had was the electron surface and the protein, and of course the electrolyte. So no other promoter or no other mediator was used. And we had this enzyme immobilized. And what is the big advantage of that? Is that instead of using a couple of, I don't know, milliliters or a lot of microliters, we only used 2 to 5 microliters for each preparation of the enzyme. And the enzyme was super stable, meaning that we could have it working like a full day, even 2 days without having any loss in the activity. So, of course, this is a membrane enzyme, so it was purified with some surfactants, and the surfactants were left on the electrode with the enzyme, and that is why this membrane protein has such a huge stability. But that was it. I mean, there was no other adding, and so the electrode surface really mimicked the physiologic donor of the enzyme. The physiologic donor was also we were also able to determine it was cytochrome C552. And the mechanism that it was in discussion for several decades and still is for us is this C iron B, meaning that iron B connects to cytochrome with the EMB3, but only the NO molecules are only attached to the iron B center. Why? And why we discard all the others? Because all the others will have an impact on the potential value of the center. Meaning if they attach one here and one here, for instance, or both in here. So as we did not see any change in the potential value of the sensor, that is the only way that this can be done. So moving to another enzyme, we have the FDH enzyme, formate dehydrogenase enzyme. This is, we also have different forms. We have the metal dependent, that it's only appears in prokaryotes, and the metal independent classes that are more widely available. So in our case, we studied these FDH from the sulfopyridosulfuricates, and they can have molybdenum or tungsten in their centers, and in our case, we have a molybdenum center. These enzymes also have a reversible enzymatic reaction, in this case from CO2 to formate and back. This is a very large enzyme and has 3 subunits. This subunit, the alpha one, is one that has the molybdenum center and where it is performed this enzymatic reaction. This is, as I said before, a highly pursued enzyme because it can convert directly the CO2 for instance, into formate in a very, very specific way with high selectivity. And the formate is a very valuable chemical, so it would be great if we could add the mitigation of CO2 together with the formation of a very nice add value compound. So in this case, we also use electrochemistry to characterize the enzyme and the catalytic activity. So in non-turnover conditions, we are able to see different processes that were due to the different redox process. Namely the clusters, the iron sulfur clusters, and the cytochrome, and in groups within the protein. But we could also see the main center, which is the made with 2 pyranobacterium cofactors, and it's here the molybdenum. And we also had the possibility to see the hydrotransfer mechanism and also all that with direct electron transfer. So here, for instance, we have catalytic conditions that we usually have in the lab. And here we have this enzyme, and we are having hydrogen carbonate in equilibrium, which gives some CO2 in solution in a very small quantity. And in this case, you see that the catalytic current is increasing with cycles and also with the presence of the hydrogen carbonate, but very, very small. So the catalytic currents are increasing just a little bit. When we pass to a saturated solution with CO2, we have a very huge catalytic reaction. And when we have direct CO2 gas injection, see for instance the scale of this one and this one, we have a much larger current, meaning that a direct CO2 injection into this protein, although the protein needs to be in aqueous solutions, it can really work with direct CO2 injection. And that is quite interesting for applications that we can see later. So also with FDH, we have tried mediated versus direct electron transfer. As you know, mediated, we use some small molecules, organic or inorganic or biological, to mediate the transfer of an electron to the protein. But we can study it, and we have studied with mediated and direct electron transfer. And in this case, although we had good results both ways, the mediated has given more nicer results. And I think in this case, the problem is really the orientation of the protein on the electrode. So if we have an orientation that allows a good electron transfer, but also allows the substrate to come in. So in that case, direct electron transfer and this type of studies are okay. But if the orientation is not the best, then small mediators, we did this with methylvalogen and also benzylvalogen, but also with cytochromes, can have an advantage in coupling with the enzyme and transfer the electrons. And so we have to also always see how the interaction with the electrodes are made with these biological species. Okay, so in this case, we assume that there is room for improvements by changing the electrode surface or interactions. So regarding our last enzyme, so I have the nickel iron hydrogenase, which is one of the hydrogenases. There are basically three. The iron that is present in prokaryotes and eukaryotes, the nickel iron prokaryotes, and a very recent class that only contains one iron that is found in archaea. So in our cases, the nickel iron hydrogenase enzyme is a very interesting one because it is highly sensitive. Although iron is highly sensitive, this one is very tolerant to some quantities of oxygen. And in that case, is also very useful to be applied in industrial processes. So this is also a periplasmic enzyme and also has two subunits. And of course, the chemical reaction is zero as it is established. So in this case, this hydrogenase, it has a lot of possibilities as the mechanism. So it has a lot of possibilities of their structure. So we have this nickel H is unready structural status, and then we have this. This is already ready, okay, and is ready when we have this conversion and or this one. And in this one, this hydroxide bridge is transforming to a nitrite bridge where hydrogen is bridging nickel 3 and iron 2. And here, this hydride, which is absolutely essential for the activation of the enzyme, and then the hydrogen is passes only for the nickel and is then, of course, released. So for this, it is essential that we have enough electrons to be sent to this active site, and also protons. Also, we have multiple redox states, but these are not only regarding the center, but also the IMS on the protein. So what is interesting is that we have a mechanism, which I'm not showing you, but you can find in the literature, that is already set for the chemical control, meaning that the chemical control of the catalysis and the electrochemical control. So the electrochemical control makes these activations through the position of potential, of course, in the presence of protons, and it gives a very straight and direct hydride bridge. And this hydride bridge can be seen by these electrochemical features. So then it can also, with the addition of a proton, it can release the hydrogen. And after releasing the hydrogen, we will get the initial, initial version of the active center. And once again, we can make the cycle like this. And this can be all controlled by the position of potential. We can also see that even if we go to a higher reducing state, it still be available to go back and produce these active states. Also, what we saw is that after an oxidation with a large amount of oxygen or with potential of 0.1 positive, we have an inactivation of the protein. And this inactivation is because we regain the oxygen in the bridge between the nickel and the iron. But when the control is just a chemical one, we have to achieve the reactivation of the enzyme, we need to impose an hydrogen-rich environment by gassing the solution and let it be among let it be together for at least 24 hours. But if we use electrochemistry, we can just cycle among a value between minus 0.6 and around 0, let's say, and we regain the initial conditions of the protein. So we can do it also by chronometry at minus 0.6, meaning that we don't really need to reactivate it with hydrogen gas or some couple of mediators and so on. We just use electrochemistry and it reactivates to the full active form. So all of this is connected with the methodology that we use. And of course, you all have heard of it. We can do it in solution, of course, but we can also do it with adsorption and with thin layer. What interests is we have to establish a very robust enzyme-electrode interface. And of course, we have to characterize electron transfer kinetics. So as I said to you previously, some redox processes within these proteins have high electron transfer concentrates and the others have low electron transfer concentrates. So we have to be careful and we have to see exactly what we want to see. And we have to establish if these processes have really a high rate of electron transfer with the electrode or the physiological couple, or high or low. And because in that case, if we have a high electron transfer concentrate, we really need to supply slow let's say, so we have to use a slow scan rate. And if we have a very high electron transfer, like for instance in the NOR, we need to supply a lot of electrons immediately, or let's say using a very high electron transfer rate, so a high scan rate that should be used. So the strategy, of course, is to see the charges in the proteins and the charge of the surface when we use these adsorption methods. Also, we can use this immobilized thin layer that when a membrane is used to retain physically the protein within the vicinity of the electrode. In this case, we consider this layer should be around maximum 100 micrometers to be sure that we are in these conditions. And in this case, we also need to be careful about the charges of the membrane that is going to influence the orientation of these proteins towards the electrode. Also, we can use this classic methodology of 1 or 2 or 3 compartment electrochemical cells and make a characterization in solution. But in this case, we can use mediators or not. But what happened is we use a lot of protein. So here, usually we have to be in the micromolar or millimolar concentration range. When here we really need small amounts and a smaller concentration of protein. So the characterization techniques usually are cyclic voltammetry, that allows us to identify the redox potentials, the reversibilities, and the different processes, and also decorrelate the process. We can use chronoperometry, meaning that we actually impulse one or more potentials with time, and we, of course, use rotating these electrodes to evaluate the mass transfer and the kinetics of the reactions. And as you know, in these conditions, the scan rate is no longer an issue because it will depend on the rotation rate of the electrode. So we also use a lot of other techniques, naturally, spectroscopic ones like EPR and UVVs. We also use electrochemical quartz crystal microbalance and also, for instance, SM and AFM for the characterization of the electrode surfaces upon the mobilization of the enzymes. So the key results that we attained, we really studied these systems for a long time, and so we have a lot of knowledge, but it was not really in the first years. It took a long time to master the systems. So, for instance, in nitric oxide reductase, we were able to optimize the conditions for the catalysis and the stability, and we are able to see the different redox transition and the catalysis. These are all pH dependent behaviors, and of course we calculated all the right constants, and we could see different behavior for NO and O2. I told you before that these enzymes are quite selective, and they are. But in the case, nitric oxide reductase can selectively catalyze NO and O2 because they are catalyzed in different centers. So the NO is catalyzed in the iron B together with the iron B3 center, and it takes the imposition of potential that is close to minus 0.6, and oxygen is catalyzed only on the B3, meaning that we can have oxygen and NO in the system, and we can selectively choose if we want the O2 reduction or the NO reduction. And they can be both or just one. And when we compare this when the enzyme is immobilized in graphite or when immobilized in solution, we can see a difference in the catalysis that we have when we have here the catalytic current or the velocity. So here we see that in the NO they are different, and in O2 they are more or less the same. And why? Because for O2, and when we use these methods in solution, we use mediators to reach the potential that is necessary for its reduction. But for NO, we really cannot reach that far, and with mediators. And so what happens is we only get there with the use of the electrons that are provided by the potential state. And this gives a very different shape in the catalysis that we get. So the key findings for formate dehydrogenase, we can really make the CO2 reduction to format at potentials that are way more positive than this. The Faraday efficiency is very high, and the activity can be enhanced in this case with mediators, but it can also work without mediators. And we can also control the catalysis. And the catalysis control is really essential for the applications. For the hydrogenase, we also attain a lot of interesting results, not only the mechanistic the processes, but also the catalysis, and we can use it directly with an electrode, or we can use it among a system that also works. The big advantage of this system for the hydrogen production. Yes?
Yes, I think that this should finish really soon. Have you got your lunch?
Sorry? Sharon?
It's interference, Cristina. You're welcome to continue. Yes,
okay,
thank you.
Sorry. I thought it was something that you said. Okay.
Yes, we have 10 minutes left, Cristina, so if you can please wrap up and we can have perhaps a couple of questions. Thank you.
Sure. So in this case, the big advantage of the nickel iron hydrogenase is that the high selectivity, so we don't really need to purify the hydrogen after. Of course, we can also use the hydrogenase as a full bacteria, and several people do that. But with the purified enzyme, this is much more a clean process, let's say, like that. So moving on very, very fast, so we can map the catalytic setters different potentials, and we can ascribe the electrotransfer pathway in each of them. And regarding applications, so in this case of the nitrogen oxides, they can be applied directly to streams of gases that have these molecules like NO, but we could also use it as for instance sensors. In case of the CO2 valorization, we can use FDH, of course, to capture and convert it, and to get added value products. Okay, so in this case, we can cut CO2 emissions and also have benefits with the circular carbon economy. Regarding the hydrogen from hydrogenase, of course, this operates in mild condition, which is really a huge advantage. It uses nickel and iron, but these are incorporated earth-abundant metals, and it's a very low quantity in the enzyme when in comparison when we use it in, for instance, electrodes in an electrolyzer. Of course, there is selectivity, already said, and is compatible with intermittent renewable energy sources because, of course, it can be, it is not really dependent on the solar production, of course. So the efficiencies are very high, and we, although there are already heterologous expression systems, we need more efficient ones in order to lower the costs and try to take advantage of this system. So just wrapping up, we have these selected publications that you can see, and more others that we can see that can give you an idea of how to study these type of systems and the details and experimental details of all of this. And the future directions, well, I would say that The future actions will be to scale up of these systems and to enhance the stability, the coupling with the renewable energy sources, and to prepare multi-enzyme cascades. We already started. This is just a funny picture, and our potential for scale up. Now we are here, but we intend to go very quickly to the medium-term goals like a pilot plant integration, and this is a long-term vision industrial implementation. These three enzymes by themselves will help a lot to reach several sustainable development goals, and I think they will be quite interesting. I hope, of course, that this were not so much boring. I'm really sorry, my English today is not on its best, but we are open to collaborations. And if you have any doubts or anything that you want to discuss more in deep, please reach up, and we'll be glad to talk to you more. Okay, thank you very much for your attention, and I'm really sorry that my English today is really not that good. I'm
No,
really
no, no.
tired, and that's what happens.
No, thank you very much, Christina, for the time you've taken to prepare this presentation, which has been enlightening of the different mechanisms that enzymes and as well the characterization of enzymes and their role in the electron transport chain. So let's give a big applause to her. And I will turn, let me just stop the recording now.
Thank you, Ashley.